Hi, I'm Mark. I'm the CEO of Saku. I'm actually a serial entrepreneur who has actually built a couple seven-figure e-commerce companies. I realized that I didn't want to just make money for the hell of making money. So I took that change, actually reinvested into getting a couple science degrees at UCLA, ended up in the lab at Dean DeCarlo, which is actually the same lab that Westbrook was in. That was about 8 years ago. And what I actually did was I took a lot of the fundamental stuff that Westbrook worked on and actually reconverted it into something that can be used for a different application. And one of the things I did learn while I was in the Carl lab is that biotech is in the middle of a revolution right now. This includes things like antibody therapeutics, gene therapy, cell therapies. But the only problem right now is that the scalability and cost issues, which I think was brought up in the last talk, actually. And the reason for this is that while there's a huge demand for a lot of these products and these products actually work, companies really can't scale to meet that demand. And the reason for this is that manufacturing costs make many of these biotech companies unprofitable. For example, in the gene therapy space, it can cost about $2 million to make an individual dose. And so while they work and can actually cure a lot of patients, a lot of these costs actually render these biotech companies unprofitable. We kind of dig into where a lot of these costs come from. And so typically they come from low cell output from these manufacturing processes. And so how these products are actually made is that you have these cells like bacteria or yeast cells or animal cells like CHO cells, for example, grow up in these tanks called bioreactors that produce this product. And ultimately what it comes down to is that they just produce very little output. And so you need huge tanks, lots of materials to actually produce a certain amount of product, which drives up cost quite dramatically. And so what we need to do is find a way to make higher producing cells to bring more products to market. Because until we can do that, a lot of these promising life-saving medicines, industrial materials, and agricultural products that people have been talking about for a really long time won't actually come to fruition. And so what we believe is that we developed one of the breakthroughs in the space called Pico shells. And what these are these microscopic particles that act as tiny test tubes. Here's what they look like. And so here on the left is a pelleted 10 million particle population. On the right is a microscopic image of these particles. And so unlike Westbrook's particles, which are completely solid, these are actually hollow. And what these do is that allows you to put individual cells into those Pico shells and allow those cells to actually grow and produce the product of interest. The way that this is going to increase manufacturing output is that you can place individual cells into these Pico shells. You can allow those cells and produce the product of interest. And then you can go ahead and isolate those highest producing cells using something called flow cytometers. You can get those top 1 in a million producers, take those cells and then scale them up in those large tanks. And so if each of those cells is producing more, then the overall output is going to increase quite dramatically reducing the cost as well. And so if we kind of compare Pico shells to some of the other technology in the space, the main ones are going to be well plates as well as microfluidic systems. So the problem with well plates is that they have relatively low throughput relative to the whole genetic diversity of cells. So your likelihood of getting that really, really good cell is going to be really low. And so if you look at microfluidics, you can get higher throughputs. But unlike with well plates, where you can actually measure based on cell productivity, the ability to measure based on cell production using microfluidics is actually very, very limited. And so with Saku, what we're able to do is maintain those throughputs that you can get with microfluidics, but it actually enables you to measure based on cell production, making it useful for this application. And so another major innovation with Pico shells is that it eliminates this lab to commercial translation issue. And so a major issue with microfluidics and well plates is that they don't replicate the environments of bioreactors. And so what ends up happening is that the cells found in the lab don't actually translate to the commercial setting. Now, the nice thing with Pico shells is that you can place them directly into bioreactors. And because those Pico shells are porous, they can get exposed to all those environmental conditions that you actually experience in the commercial setting. And what this will do is this will streamline the cell selection process to commercial use, accelerating those cell development cycles, getting those products faster to the market. And so our business model is actually not to sell the Pico shell technology. We kind of looked at other, screen tech in the space like breakthrough lights and sphere fluidics. We realized that that's not really the best way to get these screen techs to market. One, they don't generate good margins, and two, screen tech tends to be not very scalable in the form of device. And so what our plan is actually to use technology to make cell lines that are licensed out as products. And so what the idea is that we'll make a portfolio of cells, yeast cells, bacteria, CHO cells, hex cells, that will be licensed out to customers to make their products. And what those will do is this will drive down costs, making more companies profitable. And hopefully, and our mission is that this will bring biotech to scale across many industries, not just the biopharma space, but also fertilizers, foods, bioplastics, enzymes. biofuels, and many others. And so if we look at a standard cell pricing model within the industry, and so you have upfront costs, 20 to 100K, milestones, so let's say your product hits clinical trials, you can actually get royalties on these. And so that's actually really standard in the industry. And so a lot of these biopharma products can make about $200 million plus annually. And so if we can get a 2% royalty on that, that's $4 million plus annually of passive income. And that'll result in, billions to hundreds of millions of dollars TAMs for various different markets like gene therapies and protein therapeutics and industrial enzymes. And so we've already developed a lot of institutional and commercial traction. So while I was still in the lab, we had some funded projects actually with NASA and Viridos, which is a synthetic biology company right up the street. We have an overwhelming number of customers right now, including the Berkeley National Lab. We actually have a government contract being finalized right now. And actually, a couple of years ago, I got a ton of interest from a lot of major players in the industry. I realized we were going a little too fast at the time, so I actually took a step back, took everything back to stealth mode. But I think we'll be ready to serve these guys sometime next year. And so we do have a pretty amazing team with a diverse set of skills, not only in the technical realm, but also the business realm, as well as a wonderful advisory board, including Dina DiCarlo, who it seems like we have a pretty good representation here at TAE, as well as some legal and communication experts as well. So thank you. If you want more, just go to my email right here. Thank you.
Questions?
Okay. Does it dissolve, or how do you get the cells out?
Yeah, so there's two ways of doing it. So using some of the chemistries that Westbrook developed, you can actually add chemicals to them. So let's say it's a disulfide linkage, you can add DDT. It can also be a peptide. We can add an enzyme to break it down. But actually, some of the cells can just grow out of them, and so you actually don't need to add chemicals. Yeah. Yeah. Exactly.
Great talk, and interesting technology. The question I have is, are the gels porous, and how it maintains osmosis or the nutrients the cells require in order for the cells to kind of maintain the state that they want to be? That's one of the part of the production manufacturing consideration.
Yeah. So actually, it's yes, it's porous, and actually can tune the pore size. And so small molecules like nutrients and stuff will actually readily diffuse in all the time. But the unique thing is, let's say it's a secretion, you can actually tune the pore size such that you can retain that protein, for example, or some of these large molecules. You can actually also, let's say you don't want to do it that way, you can actually modify the shell such that you have capture sites that can retain secretions as well.
Can I ask something?
Yep, go ahead.
Yeah, maybe I missed something because the audio is not so good. But how do you see this innovation impacting the future of the industries?
So I think what I believe is that a lot of these cell lines are going to drastically reduce the costs of manufacturing, right? And so let's say with antibody therapies right now, they can get 10 grams per liter. Based on some of the results they're getting that I can't really talk about right now, I think we can get to something like 20 grams per liter, which will reduce the cost by 50%, right? And what that'll do is that a lot of these companies that were previously unprofitable all of a sudden become profitable. And so you have a lot more innovation in the space.
Thank you.
Sorry, I've got a question as well, if that's okay.
Go ahead.
Hi, I've got two questions actually. The first one is along the lines of your IP. You didn't mention anything about any kind of IP that has been filed. And the second, I'd really like you to explain further your competitive advantage and the market opportunity.
Yeah, and so the first question with regard to IP. So we did file IP at UCLA. We currently have an option agreement for exclusive negotiating rights with UCLA, and we're in the process of working on it right now. With regards to competitive advantage, so we're collecting some KPI data right now, but what I believe is that we'll be able to develop these cell lines way faster than everyone in the space. If you actually talk to customers, they're like, oh, I wish I had high throughput, and I wish I can solve this lab to commercial translation issue. And so what I'll do is that we will quickly outcompete all these other cell lines on the market and develop kind of the best cell lines that they'll come to us and license it out.
And your success is not going to be easily replicated.
No, because you can also, with regards to the new cell lines you can develop, you can actually get IP on that as well.
Okay, thank you.
Very good, thank you.